Hello, guys. This is going to be the start of another chapter, chapter 12, and this is where we're going to dive deep into what the DNA of a cell does, the structure of a DNA, how it replicates, how it is transcribed, then how it is translated. The first part of this chapter, we'll go over the structure of DNA and its importance. The second part will then go into the central dogma of molecular biology. That is going from DNA to RNA to a protein. Now, before we get into like the kind of the nitty gritty stuff, I do a section on, and the book does as well. I do a section on the history of DNA. I am back and forth on whether I should even include this. Every subject within biology or every chapter has a history of how it was discovered. For some reason, scientists and science educators like to really dive deep into here and show this process. I tend to more want you to understand the structure of DNA rather than the history. I can know the history. You guys can know the structure. It's in my opinion, it's a little bit irrelevant to being a scientist. Yeah, we should kind of know how things came to be, and it's kind of interesting. And it wouldn't hurt you to know this stuff, but looking at the fundamentals, I want you to know what the structure of DNA is. Now, going back, I actually on the AP Facebook group found this picture. I thought it was just amazing. I wish I had this book. I go around and I look for these kind of books in places like bookstores, and I can never find one. It's an old genetics textbook. You can see down here, this is a date, 1952. DNA is only mentioned on this one page. Can you imagine having a genetics textbook, and there's like one or two pages where DNA is explained? That's how much they didn't know about genetics at that time period. It just blows your mind that they didn't know. All right, so back in 1869, Friedrich Miescher was a scientist, and he extracted white blood cells, and he lysed them, and he found this substance in there, and he called it a nuclein. And again, this is just the first kind of identification of, hey, there's some substance here in the cells in the nucleus. Along comes Phoebus Levene in 1910, and he identified using, so again, this is like 50, 60 years later. He identified that that DNA has a substance that is made up of bases, sugars, and a phosphate group. Now, he thought they were in this kind of ring-like structure, and again, we know that it's not in this ring-like kind of structure here. But he knew that there were four nucleotides per molecule. Well, he thought there were four nucleotides. I'm sorry. He thought there were four let me go back here. Phoebus Levene in 1910, so again, about 50, 60 years later, identified that the nucleotides or the DNA was made up of bases, sugars, and phosphate groups. Now, he thought this was a ring structure that was made out of four of these nucleotides, but we know this is wrong today. But he had a good hypothesis for the data at the time and figuring things out. Died in 1940 before his contributions were really known. So again, he's another guy, took a couple steps forward. People were like, look at this guy. And then it turns out he was correct. Not about the structure of a nucleotide. I should say, not about the structure of DNA, but the structure of the nucleotide. All right. So again, 1928. There's an experiment. It's very, how should I say, famous within genetics and DNA. Griffith was working with this bacteria that caused pneumonia, and he had two different types of strains. The strain that looks smooth would kill the mouse. The R strain would not kill the mouse. You can see here this diagram. Rough strain doesn't kill the mouse. Smooth strain kills the mouse. So what he did was he boiled the S strain, killing the cells. Now, when he did that, he then injected the boiled S strain. So again, he heat shocked them, heat killed them, and it wouldn't kill the mouse, which makes sense. If we go back, this smooth strain would kill the mouse, but when we boiled it, it would destroy the cells and it would kill the cell, so it wouldn't kill the mouse. Yeah, it makes sense. Okay, that makes sense. So like again, like this would be you going out and drinking water from a stream. Yeah, you might have the chance of getting infected. If you put it over a fire for a while and kill all that bacteria, you're not going to die. So it's kind of that process. So what he did was he then mixed the heat killed smooth strain with the rough strain. Both of these shouldn't kill the mouse because again, this doesn't kill the mouse and the rough strain doesn't kill the mouse. However, when he injected both of them together, it kills the mouse, and that doesn't make sense. Okay. What he thought was there was some type of substance within the heat killed smooth strain that was being transferred into the rough strain, making it deadly. And that's what he observed. This is called the transformation principle. So again, we have the rough strain, doesn't kill the mouse. Smooth strain kills the mouse. When you heat kill the smooth strain, again, you boil it, it doesn't kill the mouse. That makes sense. When you combine these two, the two that don't kill the mouse, it kills the mouse, and that doesn't make sense. What's going on here is DNA from the smooth strain is being transferred into the R strain, making it deadly, proving that DNA has some substance that is not, I shouldn't say going to kill the mouse, but is going to be play a key role in the phenotype of the organism. Later experiments, so Griffith worked with somebody called Avery, and later experiments showed when they added, again, they did a lot of the same experiments, or she did a lot of the same experiments. They added something called DNA ACE, and that breaks down, again, it's an enzyme that breaks down DNA. When that happened when you mix them with the heat killed smooth strain and the rough strain, when you broke down the DNA, then there's nothing to be transferred into the RNA, so the mouse would live. However, when you added protease, which is an enzyme that breaks down proteins, they would not work because, again, we're not breaking down that initial DNA. It's breaking down the proteins produced after the DNA, and again, those are going to get used up at some point. So the DNA is going to keep on transferring the properties to the S strain or the R strain there and kill the mouse. I know this is really confusing. All we're doing here is we're saying, hey, DNA codes for life. And I know you're like, oh my gosh, like this is so new, and this is so, I understand this is like, this is almost like 100 years old now, but this was very monumental at the time. All right, let's see here. The early 1900s, scientists didn't know what the blueprint of life was. Like I said, this is a big stepping stone in genetics. We did know that the proteins were made of 20 different amino acids, and they had a lot of variability. They actually thought since there was 20 amino acids, the variability is much more higher. If you think about just that sequence, there's a much more. I'll just keep on calling it variability there. So that they thought that the protein actually would be the code for life. But then it turns out DNA was, and it only has 4 nucleotides, which didn't make sense. They thought, hey, this doesn't have enough variability. There's only so many ways that this can go in a sequence. Now we're going to learn later on that codons solve that, but initially scientists did think it was the protein that was code for life. Within our next experiment, we have these things called bacteriophages. Bacteriophages are a type of virus that only affects bacteria. You might have seen this on Jimmy Neutron. This was in, I think, Carl's stomach at one of the episodes. But this guy here, he actually will attach to the outside of the bacteria cell and inject the DNA inside the cell, and then it will replicate and make more of these bacteriophages. So Hershey and Chase, they were trying to figure out what was the blueprint of life, whether it was proteins or DNA. What they did was they labeled the virus outer coating, which is a protein with a radioactive isotope. Now they could watch that where that protein would go, and they actually could kind of glow a little bit. And what happened was the new viruses, when they tagged just the outer coating, were not also radioactive. When they did this same thing to the DNA, so they labeled the DNA with a isotope that was radioactive. The new viruses were also radioactive. This was evidence that the DNA is a blueprint of life and not proteins. So again, I know it's like, oh my gosh, like you're telling me DNA is the blueprint of life. I learned this in ninth grade. These scientists in the 1900s, they had no clue on this. This was fundamentally, groundbreaking for what they were researching. Let me show you, excuse me, a diagram. This does a lot better job of explaining it than I do. You can see here this is the first one where they tagged the outside, which is the protein coat. They injected their DNA inside, and then the new viruses weren't radioactive. When they radioactively tagged the DNA, you can see here they're going to inject that DNA, which is still radioactive, and then the new viruses are also going to be radioactive. This means that DNA is inherited, not the protein. Again, wow, this is crazy. DNA is passed on from parent to offspring. Like, yeah, I know. Again, it's pretty like basic for us, but back then it was big news. All right, so along in the I think the 1930s and 40s, scientists were trying to figure out what was the structure of DNA. So we know it was made of a base, a sugar, and a phosphate group, but we just didn't know what the combination was or what the structure was. So there was actually a mad kind of like a dash or like a just a push forward to try to figure out the structure of DNA because they knew whoever figured out the structure of DNA would be in every textbook from that point on. So many scientists and universities and companies were trying to figure this out. Rosalind Franklin is a scientist at this time. She was a female scientist in a very male dominated field, which means there was a lot of guys and not a lot of girls. And the guys were not particularly very, we'll say, kind to the females. Just as a side note, it is much different in STEM and science, technology, engineering, math. It is much different. There's a lot more females, and there is a lot better ethical treatment of people in these fields. And it wasn't just science that was going on, but this is, I call it like a black eye for science. Like this is a kind of moment where it's not, we look back and we're like, wow, that wasn't right. But what happened was Rosalind Franklin was taking these X-ray or diffraction photographs, and there's a lot of science behind this. I'm not going to go into this. They actually still use this process today a little bit, but they do it a lot quicker with a lot more computers. I forget how long this first image took her. It must have been like the exposure time was like 50 hours or something. That was like hours upon hours. They can do this within seconds now. It's crazy. But anyway, so she took this photograph 51. It took her so much time. And this pattern, I know it doesn't mean much to you, but you might have seen this in the back of my screen as a desktop. This was monumental. Whenever she took this photo, it was like, oh my gosh, like this is a really good photo, really good resolution here. What happened was though, Wilkins, who she was working with, not for, shared, I put it in quotation marks, shared the photo with Watson and Crick, which were two kind of mathematicians trying to figure this out. They weren't getting good data, weren't getting good pictures, but this was a monumental picture for them. They understood then what that pattern would be. And they really didn't tell Rosalind that they had stole. Like I have their share. They stole their data, and it's a kind of just a horrible moment for science because this woman who did so much for the research of DNA didn't get any of the credit for the structure of DNA. Now, I'll show you a video in class, but there's plenty of videos online. You can see here. This is Rosalind Franklin on the left, and then Watson and Crick on the right. And before we get into DNA, basically what happened was Rosalind Franklin, because of what the X-ray diffraction machine was very radioactive. It actually, if you go back to the place where she studied, it's still a little bit radioactive. She developed cancer soon afterwards and passed away before Watson and Crick won the Nobel Prize. I think it was 62 or 68, somewhere around there. Anyways, they won the Nobel Prize for figuring out the structure of DNA, but she was given no credit. And the gentleman wrote a book and were very, I'll just say, nasty and mean. They called her a nickname that she didn't like, of Rosie, and she didn't get any of the credit. You can't win a Nobel Prize post-mortem, which means after you die. And there's been a resurgence of kind of telling the real story of she did a lot of the data and research, but Watson Crick used it to figure out the structure of DNA. Now, there is the other side of the coin here is, did she understand what she was looking at when she was doing this? It's hard to tell. Would she have came to the same conclusions? Yes, she did. She actually published work on this. So it's funny though that the journal put her article second, so it made it look like she was supporting their structure, and it's not the case. They both came to the same conclusion, and again, during this time period, this is what happened. So I always like to say, yes, Watson and Crick, they are credited with the structure of DNA, but we also need to remember that Rosalind Franklin was also fundamental in this structure and the this whole moment in time. So I give Rosalind Franklin a ton of credit. And she also, I forget, she also did something with viral coats, and they think that she would have won a Nobel Prize for that as well. Anyways, just understand the history here a little bit. If it like, don't dive deep into it. Just understand, hey, these people are figuring out the DNA is the structure of life, or the fundamental blueprint of life.